Sige. So, hi, my name is Nina Girafiti Mama Ami.
Hi, my name is Vidya Mirza binti Zuhairi.
Hi, my name is Sofia binti Fadli.
Assalamualaikum warahmatullahi wabarakatuh and salam UITM hatiku everyone. On 12 June 2025, we the student of Bioforces went on a field trip to Putrajaya Wetland Park and Putrajaya Botanical Garden. Our first destination was Putrajaya Wetland Park followed by the Botanical Garden. We begin our day early in the morning after having light breakfast at Sunbreak. We gathered at the bus stop near College Seroja at 7:30 in the morning. This was our designated meeting point. Once the attendance was taken and the numbers of students was confirmed, we were divided into two buses. We departed shortly after 8 o'clock, accompanied by our lecturer, Associate Professor Dr. Faiz Apardi, Associate Professor Dr. Asmida Ismail, and one of the postgraduate student, Muhammad Hakimi bin Muhammad Nizam. The journey to Putrajaya Wetland Park took approximately 30 to 45 minutes, and we arrived there at around 9 o'clock. Upon arrival, we were given a briefing about the wetland, including its purpose in conserving biodiversity and managing floodwaters. We enjoyed listening to the session. Before starting our group activity, we were served a traditional nasi lemak breakfast that reminded us of home. After the meal, we carried out our task identifying various species such as Hagwana malayana and worked efficiently in our group. We proceed to the reference zone near the wetland to locate the assigned plant species. Although initial identification was challenging, we eventually found a specimen and recorded it on the worksheet. After verifying through online sources and consulting a field expert, we discovered it was incorrect and resumed our search. Despite the effort, the experience was both insightful and rewarding. Here are the species we find, with Elocharis dalchis and Scirpus mucornatus being the hardest for us to identify. After collecting all the required species, we proceed to the final checkpoint. Along the way, one of our sample fell and we had to turn back to retrieve it. Upon arrival, our specimens were checked by a field expert and we were fortunate that all were correctly identified. After completing the activity, we had the chance to observe swan and take group photo as a keepsake. We then had lunch, perform our prayers, and continue our visit to the Botanical Garden.
Hi everyone, my name is Vidya Mirza and I will explain about the practical one, which is basic concepts of statistics and variation. In this practical, we will learn about normal distribution and its parameters, the mean and variance, and initial understanding about correlations. The objective of this practical first is to calculate and describe the following descriptive statistics: mean, variance, and standard deviation. Second is to understand the concept of correlation, and third is to determine the relationship between two variables. In this video, I will be answering both select questions. Let's begin with question 1. We were asked to discuss the differences or similarities in the mean values of leaf length and width across the three sets of leaves. From the data, the mean leaf length for set 1 is 23.90 cm, for set 2 it is 21.94 cm, and for set 3 it is 20.35 cm. As we can see, the average length decreased from set 1 to set 3. This shows that there is variation in leaf length among the sets. The differences might be due to natural biological factors like the leaf position on the tree, sunlight exposure, or how much nutrient each leaf receives. For example, leaves that are more exposed to sunlight or located lower on the plant might grow longer due to better nutrient supply. Now, looking at leaf width, the mean for set 1 is 4.60 cm, set 2 is 3.87 cm, and set 3 is 3.74 cm. The differences here are smaller compared to leaf lengths, but we can still see a slight decrease. This shows that while leaf width is more consistent than length, it still varies between sets, again likely due to natural biological factors. To support this, we also created a scatter plot showing all 13 leaves, where we plot leaf length on the x-axis and the leaf width on the y-axis. Based on the graph, we can observe a positive trend. As leaf length increase, leaf width also tends to increase. Although all the data points don't form a perfect line, the overall pattern suggests a linear relationship. This means that longer leaves are generally wider and the two variables are positively correlated. There are few outliers, but the general direction of the data support this upward trend. Question 2, which is about variance, standard deviation, and what it means when we say the leaves have different measurement of width and length. In this experiment, variance is a statistical value that tells us how much the leaf measurements, both length and width, are differ from the average. Basically, it helps us understand how spread out the data is. If the variance is high, it means the leaf size are more varied. If the variance is low, it means most of the leaf measurements are quite close to the mean. Looking at our result, the leaf length varies was highest in set 3, which was 7.63 cm, followed by set 1 at 6.30 cm, and lowest in set 2 at 4.24 cm. This shows that leaf length in set 3 were more inconsistent or more spread out compared to the other two sets. When we say the leaves have different measurement of width and length, it simply means there is natural variation among the leaves, even though they came from the same tree. This could happen due to differences in how much sunlight each leaf receives, how much water or nutrients it absorbs, or even its position on the tree. These biological factors can influence how big or small each leaf becomes, and this is clearly reflected in our variance values. As for standard deviation, it is just the square root of the variance. It gives us a clearer idea of how far each value typically is from the mean, and the nice thing is, it is in the same unit as our measurement, which is centimeters. For example, the standard deviation for leaf length in set 3 is 2.76 cm, so that means most of the leaf lengths in that set are around 2.76 cm away from the average. In short, both variance and standard deviation helps us understand how consistent or inconsistent the leaf size are across the set, and they are very useful when comparing data like this. Question 3. I will explain why it is important to use different sets of leaf measurement in this experiment. The main reason is to make sure our results are reliable and accurate. If we only measure one set of leaves, let's say just 10 leaves, the findings may only reflect that small group and not the overall variation in leaf size. By using 3 different sets and collecting a total of 30 leaf samples, we get a better picture of the natural variation that exists even among leaves from the same tree. This includes differences in length and width that can happen because of things like sunlight, nutrient access, or leaf position. This approach helps make our statistical analysis more meaningful, especially when we are trying to observe patterns or correlations, like the one between length and width. So overall, using multiple sets allows us to make more confident and general conclusion from the data.
For the practical tour we conduct at Taman Botani Putrajaya, the objective of this practical first to understand the basic idea of using a quadrant, second to improve our observation skill, third to see the difference in how plants are spread in the quadrant, and last to guess what environmental factor might affect where and how many plants grow. So here the quadrant one, near to the bamboo tree. The species with higher distribution is Nephrolepis viscerata. The total number of individuals is 28 and the density is 1.12, and the lower is Terminalia tatapa, which is the total individual is 1 and the density is 0.04. In quadrant 2, we identify a total of 5 different plant species. Although there are multiple species, the density value for each one are quite close to each other, and it shows only a small variation. The values range between 0.04 and 0.08, which suggests that distribution of this species is relatively uniform within the quadrant. Now let's move to the post-slide question. First, definition of quadrant. Quadrant, a square of rectangular area of land randomly selected to isolate a specific section for studying and estimating the percentage of vegetation and animal presence within it. And 2 basic requirements of the quadrant technique. First, know the area of the quadrant, and 2, the number of organisms of species in the quadrant. What would be the advantages and disadvantages of using larger or smaller quadrant to survey the sedentary organism? For larger, the advantages is cover more area, more accurate, and can detect rare species. The disadvantages takes more time and might miss more detail in species pattern. For the smaller, the advantages is quicker to use and good for looking at small scale pattern, and disadvantages might not show full diversity and can miss rare species. So, regarding the question, ecologists face several problems when doing sampling in large or difficult habitat like forest or aquatic area. One main issue is that it's hard to see or find the organism, especially if they are small, hidden, and moving around. This makes it difficult to count them accurately. Another problem is that some species are not spread out evenly. They may be found in groves or patches, so a small sample might not show the true number in the work area. And lastly, large habitats are hard to cover completely, and this can affect how accurate the average density results are. Because of this issue, it becomes challenging for ecologists to get result estimate of species distribution and population size. For the last question, the hypothesis is that this difference is caused by variation in microhabitat conditions at each size. The main factors that affect plant distribution include like intensity, soil type, moisture level, soil composition, human or animal disturbance, adaptability of the plant species, and competition between species. For example, in Site 1, Nephrolepis viscerata was found in higher numbers. This was because it better adapted to the condition there, or it grows and spread faster. Meanwhile, Site 2 had fewer species, possibly because the conditions are less suitable or the plants they are more sensitive or slower to reproduce. In conclusion, both biotic and abiotic factors influence the plant series found in each quadrant.
Fieldtrip Bio 464.
It's a wrap!